Allah, the most beneficent and merciful. Here I am, Muhammad Jamal Khan, lecturer in botany, University of Education, Lahore. Dear students, Assalamu alaikum. Today we will discuss a topic that related to our elective subject, that is microbiology, and the course code of this subject is BOTN 4128. So today in this lecture our focus is on types of light or compound microscope. So here we have types of microscope. Light microscope, one is bright field microscope, the other one is dark field microscope. Then we have a fluorescence microscope, and then we have a phase contrast microscope. In this lecture, I'll give you a very brief introduction of bright field microscope, because in our previous lecture, lecture number 5, we discussed this microscope, in which we discussed general light compound microscope. Light compound microscope is also called as bright field microscope. So in this lecture, we have focus only on next two types of light microscope, that is dark field microscope and inflorescence microscope. So first of all, we have a very brief introduction regarding bright field microscope. So here we have a index in which we discuss the bright field microscope, its definition, and then we discuss general account. After that we discuss the dark field microscopy, and in the end we discuss fluorescence microscopy. So here we have a simple light field microscope, so or bright field microscope. So the ordinary microscope or general light compound microscope is called as bright microscope, and we call it as bright field microscope because it forms dark image against bright background. In this microscope there are some limiting factors. Number one is the limiting factor is resolving power, because the magnification of light microscope depend upon resolving power, and resolving power depend upon the wavelength of illuminating light or beat. So this resolution is determined by certain physical attributes or characteristics of the wavelength of light and light generating power of the objective and condenser lens. So here we have a very simple principle of its working. Here it's a source of light. This one is a source of light. This source of light then moves towards condenser. Condenser condense this light and put it on a specimen. Here we have a specimen. After transmit from this specimen, it enter into the objective, and this objective lens then put this light to a projector lens, and projector lens ultimately form an image at an eyepiece. So here we attain a final image. So here it's a very basic or simple principle of bright field microscope. So as far as we concerned with its advantages, basically we use this microscope only for immobile specimen, bacteria, cell or tissue. So most of the transparent or colorless specimen, one thing that must be obliged is that the colorless or transparent specimen must be stained for their clear vision or for their clear image. So for this purpose we stained our specimen to find out a clear image. So this is all about a brief description of bright field microscope. So now our focus is dark field microscope. So dark field microscopy is the type of light or compound microscopy. So in this technique, we used to observe unstained sample, causing them to appear brightly against a dark background. So here, this dark field microscope is antagonistic to bright field microscope, or the phenomena of dark field microscope is totally inversely to the light field microscope, because here the image is appear on a dark background. Simply, as you observe in this figure, this is the dark background, and here we observe the image. So it means that a dark field microscope is frequently performed on same microscope on which bright field microscope is performed. But instead of the normal condenser, it contains an opaque. Here we have opaque condenser, this one. Opaque condenser that contains an opaque disc. So it means instead of normal condenser, it contains a opaque disc. So from a disc blocks like a light that would enter the objective lens directly. Here you clearly observe these are the objective apertures that block this light. Here you observe, and light only transmits through specimen. So we obtained a cone of light where rays are diffracted, reflected, and or refracted off the object, ultimately allowing the individual wave to view, or allowing the individual to view a specimen in dark field. So simply, light that has been reflected or refracted by the specimen forms the image. So the field surrounding specimen appears dark while the object appears brightly. So here we have a working principle of dark field microscope. Simply, it performed on the same microscope on which bright field microscope is performed, and in addition to this, instead of normal condenser, a dark field opaque disc or a dark field microscope uses a dark field condenser that contains opaque disc. So this disc basically blocks light that would enter the objective lens directly. So only light that is reflected of the specimen enter the objective lens only. So here we have a condenser of a dark field microscope, as you observe in this diagram. So here we have the simple principle. Firstly, in a simple light microscope or bright field microscope, this one figures indicate the light field microscope, and this indicate a bright field microscope. So in light field microscope, only the source of light entered through a condenser, then this light enter towards specimen, transmit through it, and from objective it enter through the ocular or eyepiece. But here, in this case, in front of light source there is an opaque disc. So the distract light are blocked by this opaque disc. Then light enter into a condenser lens. From this light enter or transmit through specimen, and from this only light refracted by the specimen captured by the objective lens, while the scattered light doesn't capture by this objective lens, and only the light that transmits through specimen enter to this objective lens, and after this image is formed at objective, at eyepiece, or at ocular lens. So simply we have a addition of opaque disc in dark field microscopy. So here we have another figure that's simply indicate the differentiation between bright field and dark field microscopy. Here A shows the light or bright field microscopy, while B indicates dark field microscopy. So in A, the condenser of the bright field microscopy concentrate and transmit light directly through specimen. From here and from here, all the light transmit or concentrate directly through specimen, while in dark field, condenser deflects light rays so that they reflect off the specimen at an angle before they are collected and focusing into an image. So you observe in this case, light enter through this. So light enter through diaphragm and approaches to a condenser. Here the condenser deflects the light towards left and towards right side, and after this they reflect off the specimen at different angles. These are the different angles from this different angles. So after reflect off the specimen at a different angle, so only the light approach or transmit to this specific specimen. So the light that approaches to this specific specimen gives the image at an eyepiece or ocular side. So here we have a different view related to bright field microscopy and dark field microscopy. In bright field microscopy we have a bright background, but in dark field microscopy we have a black background. Here you observe, or dark background. So this dark field microscopy basically give various benefit as compared to bright field microscopy. So dark field microscopy also become very effective as it describes microscopy methods in both light and electron microscopy, so which exclude the unscattered beam from the image. So as a result, the field around the specimen where there is no specimen to scatter the beam is generally appeared dark. So briefly we can say that that the dark field condenser with a central circular stop which illuminates the object with the cone of light, and this cone is produced due to the diffraction, refraction or reflection. So it's the most essential part of the dark ground microscope. This microscope use reflected light instead of transmitted light that is used in the ordinary bright field microscope. So it prevents light from falling directly on the objective lens. So the light rays falling on the object are reflected or scattered onto the objective lens, with the result that the microorganisms appear brightly stained against a dark micro background. So here we have different uses of dark field microscope. Basically, we use dark field microscope to view the bacteria or stained bacteria, and it is also very useful for clinical purpose. Different types of bacteria that cause disease can be viewed through this dark field microscope. So we can also observe the motility of flagellated bacteria, the movement of flagellated bacteria through this microscope. In addition to this, we observe different types of crystals, polymers, marines from this dark field microscopy, and different types of tissues and cells that are mounted on a slide can also be viewed through this dark field microscope. And it also be very helpful to examine external detail of various microorganisms. So dark field microscopy is very effective to observe different types of biological samples. Not only we restrict it to itself to a bacteria, but bacteria that is present in saliva also has been observed through this dark field microscope. So dark field microscope basically divert illumination and light rays, making detail of the specimen appear luminous. So it is very impressive and very productive result can be gained from this microscope. So we obtained this microscope provide very good results, especially through the examination of various biological samples like blood. So it can yield high magnification of different bacterial cells and low magnification of tissues and cells of certain organisms. Through this microscope, certain bacteria and fungi can be studied with by using its principal working. So the use of dark field microscope is very simple. There is no complexity or and free of artifacts, or you can say that free from complications. So very simple in use, and most of the researchers can easily use this microscope, and they want to use this microscope for the study of various microorganisms. So here we have some of the limitations or disadvantages of dark field microscope. Firstly, the light level or light intensity is very low in this dark field microscope. So the final image exhibit low light level that cause distortion. The second one is that sometimes the specimen or dust and other particles scatters the light and are easily observed through ocular or through eyepiece. Sample must be very strongly illuminated, so it may cause damage to that sample. And the last one is that we can't study the internal structure of organism through this dark field microscope, because it in this microscope light passes around the specimen rather than through the specimen. So next type of light compound microscope is fluorescence microscope. So now here we discuss the fluorescence microscope. So simply the fluorescence microscope. So in this fluorescence microscope, one have the advantage of fluorescence. So a short wavelength of light absorbed by the specimen, and in the result of this, the specimen give off light at a longer wavelength that can easily visible. So here we understand this by that if tissues, cells or bacteria are stained with the fluorescent dye and are examined under microscope with ultraviolet light instead of ordinary visible light, they become luminous and are seen as bright object against dark background. So we can say that in this fluorescence microscopy, various fluorescent dyes are used which give property of fluorescence to only a specific part of the cell, and hence it can be focused for study or for clear observation. So as a result, the color that the cells will appear depends on the type of dye and the light filters. So the fluorescence microscope is used to observe cells or other materials that are either naturally fluorescent or have been stained or tagged with fluorescent dye. So here we have a fluorescence microscope. Here we have a triangular head, two binocular, then we have a third one for camera. These are different objectives. Yes, here we have a mechanical stage. Then this is the main plant, body and power supply of fluorescence microscope. So fluorescence microscope is simple optical or light compound microscope that uses various fluorescence instead of or in addition to reflection and absorption to study properties of organic and inorganic substances. So we use fluorescence for the study of organic and inorganic substances. So this microscope or this fluorescence microscopy was devised in early 20th century by August Kohler, Karl Reichert and Heinrich Lehmann. So here we have the basic principle of fluorescence microscope. So simply the principle is that stained the component with fluorescent dye so that we can observe the specific part of the specimen. So by doing this, certain compounds are illuminated with high energy light. So they then emit light of a different and lower frequency. So this effect is known as fluorescence. So often specimens show their own characteristic autofluorescence image based on their chemical makeup. So here the component of interest in the specimen is specifically labeled with a fluorescent molecule that is called as fluorophore. So the specimen is illuminated with the light of a specific wavelength which is absorbed by the fluorophore, causing them to emit longer wavelength of light. So typical component of fluorescence of microscope are the light source. They depends upon different source. It may be a xenon arc lamp or mercury vapor lamp, and the excitation filter, the dichroic mirror and the emission filter. So these are the components of fluorescence microscope on the behalf of which this microscope operates. Similarly, we discuss later one by one all these components. Illumination light is separated from much weaker emitted fluorescence through the use of an emission filter, and the filter and the dichroic mirror are chosen to match the spectral excitation and emission characteristics of fluorophore used to label for specimen. So in this manner, a single fluorophore is imaged at a time, and multicolor images of several fluorophores must be composed by combining several single color images. So now we discuss all here. So we have two forms of fluorescence microscope. One is epifluorescence. Later we discuss in detail in our later lectures. So now we focus on parts of fluorescence microscope. So first of all, here we have a fluorescent dyes that we used or apply to our specimen. So a fluorophore is a fluorescent chemical compound that can emit light upon light excitation. So contains several combined aromatic compounds or planes or cyclic molecules or with several sigma bonds, and many fluorescent stains have been designed for a range of various biological molecules. So here we have some of the fluorescent dyes. Some of these are small molecules that are intrinsically fluorescent and bind a biological molecule of interest. So major example of these in fluorescent dyes are nucleic acid stains like DAPI and Hosch phalloidin, which is used to stain actin fibers in mammalian cells. So a light source, that's very necessary and the important part of fluorescent microscope. So four main types of light source are used, including xenon, arc lamp or mercury vapor lamp with excitation filter, laser and high power LEDs. So here we have an excitation filter. So here we have a exciter filter. So this exciter filter is typically band pass filter that passes only the wavelength absorbed by the fluorophore, so thus minimizing the excitation of other sources of fluorescence and blocking excitation light in fluorescence emission band. So the fourth one is dichroic mirror. This is also the part of fluorescent microscope. So here we have a dichroic mirror. So dichroic mirror or filter or a thin film filter is a very accurate color filter, and it is used to selectively pass light of small range of colors while reflecting the other colors. So the next one is emission filter, fifth one. So the emission filter actually is an emitter that typically a band pass filter that passes only the wavelength emitted by a fluorophore and block all undesired light outside this band, especially the excitation light. So by blocking unwanted excitation energy or light, sample and the system autofluorescence or optical filters ensure the darkest background. So here we have the emission filter. So firstly, this is the light source. Light in move towards this excitation filter, and after here light reflects from dichroic filter to objective lens, and from this it transmit or put on a specimen, and this specimen after absorbing this light, so this specimen have some fluorescent dyes. From this the light directly pass through the tube and ultimately reaches towards emission filter, and it from this emission filter it filters the distracted lights, and from this it directly move towards detector. So here we have for some application of fluorescence microscope. First of all, we use this microscope for identify various structures or various organic and inorganic substances with the help of different fluorescent dyes. So fluorescent microscope help to identify the structure of different chemical substances. So in addition to this, the next generally we use this for different scientific purpose, for identification of substances, for finding out the compound that is present within a specific substance. So in various research areas we can use this fluorescent microscope. So next we have the next application of this fluorescent microscope is the fluorescent antibody technique. So by using this technique we can find out the activity, or we can find out the emphasization of different antibodies with respect to their antigens. So for antibody studies, fluorescent microscope is one of the basic item that needs by the different scientists for antibodies studies. So basically, one particular powerful method, that is the combination of antibodies coupled to a fluorochrome as in immunostaining technique. So fluorescent microscopy is a very critical tool for academic and for pharmaceutical pathology and clinical medicine. So here I give the fluorescent antibody technique phenomena. The principal use of fluorescence microscopy is a diagnostic technique called fluorescent antibody technique or immunofluorescence, employed for detection of antigen direct fluorescent antibody techniques and antibodies indirect fluorescent antibodies method. So here we have some of the advantages of this technique. We can image the living cell through this microscopy, or even we isolate the individual protein and we observe the general structure or chemical substances of that protein. We can detect a very minute size of substances that are in micrometer through fluorescent microscopy. So here we have some of the limitation of this microscopy. First we have a photo bleaching. Basically the photo bleaching is a process or phenomena when a fluorophore loses its fluorescence due to damage induced by light. So this leads to loss of fluorescence and signal while imaging a sample. So in this, when light of appropriate wavelength is directed on a fluorophore, its transition from ground state to excited singlet and triplet stages. In its excited state, it may interact with other molecules and undergo permanent covalent modification. So based on these parameters, some fluorophores can bleach quickly, while others can undergo several thousands of cycles of excitation and emission before bleaching. So the second one is phytotoxicity. So in the phototoxicity frequently occurs during fluorescence microscopy, and its consequences are often underestimated because it causes damage to cellular macromolecules upon excitation, light illumination, and can impair or destroy the sample physiology and even led to the sample death. So the last limitation of fluorescence microscope is that it allows only a specific structure for vision or can be seen through fluorescent microscope. So all structures are can't be covered by fluorescence microscope. So only a specific structure or chemical substances that relate to organic or inorganic substances we can observe through this microscope. So these are the references that assist me in the preparation of this lecture.
I hope this lecture is very informative for you. Thank you.